morning. Good morning, NER community, NER family, NER friends. My name is Martine de Kok. I'm a computer science professor at the University of Washington, Tacoma, and I have the unique honor and pleasure today of introducing the first keynote speaker, Dr. Jean Yo. Dr. Yo is a computational biologist with a very impressive resume, with a very long list of achievements that, when you see it, make you want to double check, like, is this really all the same person? Was this all accomplished by the same person? Usually, I speak without notes, but today I thought it would be wise to bring some notes just to make sure that I mention at least all the important things. Dr. Yo is the Sanford Stem Cell Institute Endowed Chair and Professor of Cellular and Molecular Medicine at the University of California, San Diego. He is the Director of UC San Diego Center for RNA Technologies and Therapeutics. He is Chief Scientist for Sanford Laboratories for Innovative Medicine and also the Director for the Sanford Stem Cells Institute's Innovative Center. Dr. Yo trained as a computational biologist in AI during his PhD at MIT, and he studied stem cell biology as a Crick Jacobs Fellow at the Salk Institute. His lab at UC San Diego has developed some of the most popular molecular and computational technologies to study how RNA processing is affected by RNA-binding proteins in development and disease. Dr. Yo has authored around 300 peer-reviewed publications, and he is among the top 1% most highly cited scientists. Dr. Yo received many prominent academic awards. I'm not going to give you the entire list because we need to leave some time for his actual talk, right? So I'll just mention a few. He has received the San Diego Exconomy Award for Big Idea, the Paul Allen Distinguished Investigator Award, the RNA Society's Early Career Award, and the Eliza Isoralde Award for Innovation in Research, Teaching, and Service. On top of all of this, Dr. Yo is also a very successful entrepreneur. He has founded multiple biotech companies, and he serves as scientific advisor to companies such as In Vitro and Orbital Therapeutics and many others, some of which have been acquired by or are associated with Bristol Myers Squibb. That's a company I want to mention explicitly because it's a very big pharmaceutical known company, but it also means something to me on a personal level because I have a relative who has cancer and who is alive today because of medicine produced by this company. So I want to mention it to stress here that the kind of research that Dr. Yo does, it's not just about academic research and about papers and about businesses, but it's actually also about literally keeping people alive and improving quality of life. Dr. Yo is among the top 50 life science academic entrepreneurs by BioSpeaker list, and he also received the inaugural Entrepreneurial Achievement Award from the UC San Diego Rady School of Management. Pretty much all of these achievements were accomplished before we had ChatGPT, before we had Cloud Code, before we had any of your favorite AI, and before we had the NER pilot, before we had the NER, right? So we can only imagine what the future will bring. And with that, I would like to give the floor to Dr. Yo, so please join me in giving him a warm welcome, a warm round of applause as he comes on stage.
Thank you, Martina. Thank you so much. All right, thank you, Martina, for the kind introduction. And actually, she's not correct. Most of my accomplishments are not mine, they're my team, right? So I have a very, very good team, actually a couple of sets of teams in San Diego that are really, an amazing crew of people. All right, so what I thought I'll do today is tell you a little bit about RNA biology, RNA therapeutics, how we're leveraging the NER resources. So thank you, NER, Katie, and the organizers for letting us play in this space. Okay, let me see if this slides work here. Okay, so can you still hear me if I walk? Okay, so RNA today is a gold mine for therapeutics, okay? Why is that? Because when we sequence with next generation sequencing, lots and lots of RNAs in every single cell, in every cancer tissue, in every neurological diseases, we identify isoforms, circular RNAs, link RNAs, microRNAs, many isoforms, many RNA species that expand the druggable genome from the small number of druggable proteins to, by several orders of magnitude, to hundreds and thousands of RNA species. But RNA is also an interesting molecule because it's both programmable and druggable. What does programmable mean? Well, it means that you can design at will, which is great for, in the past, vaccines, now in vivo CAR T for autoimmune diseases, for protein replacements in cystic fibrosis, and for generating new antibodies. But RNA is also druggable. You can develop small interfering RNAs or antisense oligonucleotides. There are basically 18 to 22 base sequences that can recognize RNA. And by doing so, you have ushered in the next generation of precision medicine, right? Because given a sequence, given a patient mutation, you can design an oligonucleotide to affect it. And I'll show you one example. And so in my lab, we think a lot about RNA biology as a sort of a bridge, right? Thinking about basic and also translation. And so this is the typical central dogma that we see in molecular biology textbooks, right? So genes in the genome are transcribed into RNA species. And then these RNA species or intermediates that are called, right, which are not really true, right, are then translated into protein. Now, I just wanted to set everybody back to ground zero and correct this picture, right? So this is by ChatGPT, right? So here. So RNA is never naked. So we always see this string of just letters. And the field at large would always present it as just a string of letters. But RNA is never naked, right? RNA always is clothed by proteins that bind it. So we call that RNA-binding proteins, right? And I'm going to try to convince you today that this is an interesting perspective of how we think about RNA biology and RNA therapeutics. So a fifth of the human proteome consists of these proteins that are predicted to bind RNA. And this is interesting, right? Because why do you have so many proteins that have sequence specificity or structural specificity for binding RNA? We don't really know, right? And so maybe some insight can be gained from understanding what they all do. So these RNA-binding proteins modulate every single step of the RNA life cycle, from splicing, so generating different isoforms, to translation, stability, and export, and so on. So basically, as RNA is generated, it goes through sort of dynamics and processing that generate the final product that goes into the cytoplasm, and then the final product gets translated into protein. And if you think about mRNA as a drug, right, your synthetic RNAs and lipid nanoparticles are injected into you. That mRNA goes into the cytoplasm directly, and then that's translated into protein. But as soon as they get in, they're bound by these RNA-binding proteins. So RNA-binding proteins, each of these can do many different things, so I won't sort of go over all the things that they can do. So in my group, we think about this RNA-binding protein, RNA biology as a bridge to thinking about RNA therapeutics, right? Now you can design mRNA medicines at will. You can use ASOs and siRNAs to target RNAs, taking into account the RBP RNA landscape. And you can design small molecules to bind RNA or RNA-binding proteins. And I've been fortunate that I've been able to co-found some of the companies in this space that use this understanding to develop next-generation therapeutics. Okay, in fact, for those that are familiar with genome editing, base editing, CRISPR-Cas9, that is simply a RNA-binding protein, the Cas protein, guided by a sgRNA, a guide to a DNA locus for editing. For those that are familiar with antibiotics, these small molecules engage protein-RNA interactions with the ribosome. So again, perspectives like this is quite important for us to consider in thinking about the RNA space. Okay, and there's some more examples, right? So here is, as you're familiar with the mRNA as a drug, right? So the mRNA vaccines. And recently, there was a personalized genome editing therapy developed on a child with inborn metabolism disorder called KJ, right? And then, so that's based on RNA delivered using, base editors delivered using RNA. Of course, you also have siRNA therapeutics now that have been leveraged for cardiovascular metabolic diseases, and antisense oligonucleotides, which I'll spend a bit more time on, that are sequence-specific designs that can hit any RNA molecule to modify and modulate expression. So a bit on the ASOs, right? So this is a spinal muscular atrophy. It's a devastating motor neuron disease, and patients, when they're young, they have a loss, they're caused by a loss of SMN1. It's a gene that's important for this disease. It turns out that in the humans, we have a backup copy called SMN2, and if only you could generate protein from SMN2, then you have replaced the missing SMN protein, therefore fixing the disease. Now, the only difference is that in SMN2, there's an exon, right, that is misspliced. So to basically cause it to splice properly, Adrian Craner at Cold Spring Harbor, Biogen, and Ionis developed one single ASO to block one single RNA binding protein, A1 in green, from binding one single nucleotide in an intron. So that's of 1,000 RBPs on their RNA sequence that they can site-specifically design, and this is a drug, right? So this is a billion-dollar drug that cures spinal muscular atrophy, the first oligonucleotide drug for neurological disorders. So in my group, we think about if only you have a large data set where every single RNA molecule is organized, and we organize it by all the RBPs that bind it. So then you have a landscape of interactions, and we know for each nucleotide in the RNA what it does to gene expression control, splicing, export, and so on. Then you can just design an ASO eventually just by looking at the sequence, and that can modify disease. So we spent a lot of time developing ways to identify this, generate this data, this map, and some of these maps are being used for large language models and so on. So how do we identify from a milieu of RNAs, all the different RBPs that bind it, the green ones that bind specific places? So we've developed technologies in the space, AI methods to predict protein-protein interactions, protein-RNA interactions, and these have been all used by the community for thousands of labs and companies, and some of our technologies have gone into Eclipse Bio since 2017 that service and work for all the pharma companies and a large number of academic labs, and essentially all the RNA therapeutic companies to date. So I'm a co-founder of Eclipse. So today, this sets the stage for how we think about RNA biology, how we think about making impact in this space. So I'll tell you two short stories and how we're leveraging the NAIR resources and the NAIR partnership. Okay, the first one is a very basic biology story, right? I hope you are, can be interested in this, but it's a basic neuroscience story, and so I'll explain in some detail. And then the second one, it's a more translational story, meaning we are trying to develop large language models there that can be leveraged by the mRNA medicines community. Okay, so the first one is led by Karen May, an amazing postdoc now project scientist in the lab. We have many collaborators, Leon Bergen, Gentry Patrick, Trey Eidecker, and lots of folks in different labs that contributed to this first story. So I just wanted to put this out there. Okay, so let's step back again. So so this image caught my attention in 2007. This is an image from Eric Lucuy's lab in Montreal when he was a postdoc. He took Drosophila embryos, thousands of these, and layered them one at a time, and took an image with a single molecule resolution for each RNA species in the fruit fly. Okay? So you think simple organism, embryos, hasn't even developed into a Drosophila yet. Where should all the RNAs go in all the cells? It should be all the same. Turns out every single one is different. So this image here from Eric, I think, made the cover of Cell and has to this date been cited to go, RNAs have an amazing repertoire for where they are localized in different tissues and different cells. And so we are particularly interested in where RNAs are in the brain. So what's critical for every neuronal function in your body is the ability for neurons to synapse to another neuron or to other non-neuronal cells, glial cells, oligodendrocytes, right? And this is called a synapse, okay? So it transmits electrical signals or chemical signals to enable you to move, to eat, to be able to have thought, learning, and memory. But to this date, we still don't really know how all these synapses work. Why is that? Well, Some of them are 1 meter away, right, from your brain, right, to your toes, 1 meter away. And so if you have if you feel a sensation and you want to make a movement, you do it pretty quickly, right? You don't wait for 1 day for the RNAs to be made in the nucleus in the brain, and then for the RNAs to, eventually travel to the toe, right? And so the way this works is that all the RNAs are already made prepackaged. They are, well, some subset, and they've been moved and localized and chaperoned to different parts of the neuron, to different parts of the brain, different parts of the neurons, right, at the synapses. And then when you need it, then they get translated into protein, right? And so this idea of like the localization grammar of mRNAs to synapses is like a fundamental thing that's been chasing for, the field's been chasing for 20, 30 years, right? What governs this? And this is critical because in almost all neurological diseases, Parkinson's, Alzheimer's disease, ALS, FTD, frontotemporal dementia, there's always defects in synapse biology, and the RNAs that are found in synapses are always altered. Okay, so about several years ago now, Karen said, now that AI is getting to be more cost-effective, right? Can we generate this map, RNA localization map, to really understand where neurons go to synapses and how they're localized there? So this is called the RNA grammar, and we think of this as, as trying to first identify all the RNAs that are there, and then a simple problem is, can you then, given the sequence in the genome, just the transcriptome, I guess, the RNA, and go, okay, this one is going to be in the synapse at this point, like without anything else but just a sequence. And so first, this is not a new problem. So others in the field have tried to do this with less refined methods, right? Meaning start with RNAs that are found in synapses, and then go, what are the short motifs that are present in the 3' UTR of the RNA? So basically very focused looks for very, very short things. And so the context window of how we look at gene expression regulation has historically been very small, 7 bases, 8 bases, 10 bases. And this was partly due to compute, right? Okay, so on that note, right, so we think there's a need for new mRNA large language models to just even model RNAs that are in the body, but particularly in the brain. And why is this? Because RNAs in the brain are several times longer than RNAs anywhere else. And we can go into why later, right? But the existing large language models, as you can see, RNA Renamo and RNA BERT, are very small context windows, 400 bases and so on. So that covers a very small fraction of all the RNAs that are in the brain. Whereas our model has 8 kb context windows, should capture all RNAs, as many RNAs, much more, right, in the brain. Okay, so I won't go into too much detail, but basically what we've been doing is generating data from mouse brains, so synapses specifically, so isolating synapses, sequencing RNAs in there, and then making sure that they're relevant to a human system. So we take human iPS-induced pluripotent stem cells from wild-type individuals. We make neurons from that. We do the same synapse RNA isolation. That all forms large data sets that we use for fine-tuning our mRNA large language model. Then this model then gives us the ability to learn biology, which I'll explain in the next few slides, and then make predictions. So a bit more about this large language model. So this was generated from 108 mRNA from all these different species. There's about 5 million or so sequences. This is not sequencing data. This is just the actual gene, the sequence that encodes the messenger RNA in the genomes. There are many parameters in this large language model. We use the hyena, maybe it's the next hyena operator here. We have a very simple feed-forward neural network to basically build and pre-train this large language model. And then our synapse localized RNA just forms a beautiful fine-tuning data set, right? We think this is really important to be able to teach these models like the right places to pay attention to. And so we built this, and then we fine-tune it, and then we go, how well does it predict on how that data sets and all these sort of standard AI machine learning metrics, right? So these are receiver operating curves to tell you accuracy and false discovery rate balances. the higher you go on the top right, the better the performance of the algorithms. And you can see that this large language model we call Helm RNA really accurately predicts, given an RNA, given a sequence, right? Oh, it belongs to the synapse or not. So it seems like we have, with this large context window, we have cracked the code, but we don't understand what that means, right? So it does well in mouse tissue on the left, and then human iPS-derived neurons on the right. Okay. Again, if you think about the sequences that are captured, that are being, that we can run predictions on, they consist of 90-something% of all the RNAs that are they're encoded in the genome, that are expressed in the brain, because we have these large context windows. And if you run other algorithms with different context windows and different architectures, our Helm RNA on the right, on green, still outperforms all of this in terms of accuracy. Now, how do you validate thousands of predictions? And so most of the large language model papers that are published would build a billion parameter thing, and then they test one thing, right? And so we wanted to say, now we have thousands of predictions, how do we test all of these at scale? So we leverage the spatial transcriptomics platform from 10x Genomics called the Xenium platform, where you can do single molecule measurements for 5,000 RNAs in situ in the brain. And turns out Karen spent a lot of time optimizing the localization of these RNA images and also the protein images when she's staining for the synapses. So just to cut long story short here, we were able to register these images and then take each RNA and ask, is it able to be predicted by the algorithm, and does it show up at the synapse? And the answer is yes. And of course, this was, I think more than 80% of them are accurate. I don't need to sort of check that. I don't have that slide. But if you look at individual transcripts that are new, that have never been shown to be in the synapse, you can see that they're co-localizing outside the nucleus, which is this gray bubble, and next to these little puncta. Okay, so now that you've built this large language model and fine-tune it for synaptic localized RNA, can you learn something about the biology of it, right? So we do a simple thing called ablation mapping, we call, which is basically you just remove sequences, nucleotides, even one base at a time from your RNA sequences, and ask how that changes the large language model predictions, right? If you have a score that drops, then we go, maybe that sequence is really important for the model to pay attention to, which predicts its biology. And it turns out that when you find these set of motifs or now sequences within RNAs, one surprise was that it was everywhere. It wasn't just in the UTR as expected, but could be across the entire transcript. They are much more evolutionarily conserved across different species, which suggests that they are likely to be functional. That's across different regions. And then there's a database called ClinVar, which contains genetic mutations found in lots of patients with different diseases. And the diseases are mapped, right? These are genetic diseases, they're mapped, and they're annotated there. And so we basically find that our RNA localization motifs overlap within these sequences, overlap very, very nicely with patient variants, and particularly on the top right with neurological diseases, epileptic seizures, and so on and so on. So that wasn't, we didn't train that way, but that was the outcome. And then finally, we did, go back in and identify the RNA-binding proteins that bound these RNAs and were responsible for moving these RNAs back and forth. One of them is this muscle-blind protein. I don't have time to go over here, but we were able to knock it down and show that it affected the localization of many of these RNAs. And so back to the RBPs, RNA-binding proteins controlling transport. And then, of course, we also identified completely new motifs, right? Where to show that it actually worked to affect transport, we would cut it out from the RNA, make it in vitro, put it transfected into a neuron, and show that it affected localization. So you can see, I think here in this RNA, they're localized, right? Little red dots. I don't have a laser pointer. They're localized on the on the top right. But then when you cut out a single chunk of it, then it's no longer localized. It's now a blob in red. And so just to summarize this part of it, right? We think that this synaptic transcriptome measurements is a really great way to generate data sets for fine-tuning. We generated a large language model with mRNA sequences. And now we validated this to I think a large degree. And we're now looking for the right RBPs that control RNA localization. And then I guess all of this can be re-evaluated in the context of iPS cells from different neurological diseases, FTD, ALS, and so on. Okay, so then how are we extending this with NAIR support resource? So now we are basically going into looking not just at the messenger RNA, so I didn't tell you, but messenger RNAs is a small chunk of the entire pre-messenger RNA, which contains very long intronic sequences. And many mutations actually don't fall in the mRNA, which is the coding sequence, but in the non-coding intronic sequences. So why is that, right? And so we're hoping to build a new large language model that unifies pre-messenger RNA, the full-length sequence with the mRNA sequence. Yeah, I sort of mentioned this, right? And so the idea is that eventually can you go to all the point mutations, the majority of which don't fall in a coding region in the mRNA, but in the non-coding region, and ask how does it affect RNA life cycle progression. Okay, so I'm going to transition to the second part of my talk, which is a bit more less on basic biology, but thinking about how can we develop something with utility in the mRNA medicine space. Actually, let me grab a drink. Okay, but before I do that, I just want to say I wear another hat. Besides being a faculty at UCSD, I'm also the chief scientist of Sanford Laboratories. And this is our effort in trying to bridge the gap between academic research and commercial asset building and new co-formation, right? So Sanford Labs is an independent, not-for-profit entity that we've raised a fair bit of resources for in San Diego, where the goal here is to take projects that have sort of outlasted academic federal foundation funding that is moving from a basic and just needs another couple of years to get to a proof of concept point where then we can build a partnership with pharma or spin something out and so on. And so what's unique about Sanford Labs is that we have a core set of talented individuals that work in different spokes, but they all are working together. So in some biotech accelerators, you have 20 companies, they all are doing very different things and no one actually talks, right? So here we're trying to build a collection of a few companies, but they're all kind of working together, right? They share resources, they share compute as well. And so Christina Verri is the president. I just recently helped found this a couple of years ago. Okay, so right now it's all primarily RNA therapeutics focus. Okay, so and one of the legs there, right, is mRNA medicines, right? AI and mRNA medicines. So again, I maybe don't have to explain this again, right? Capstan builds in vivo CAR T. So when you want to build CAR T in the past, it would be millions of dollars to take out someone's electrophoresis someone's cells and then insert the CAR, put it back in the person for treatment. Now you can do in vivo CAR T where you use mRNA medicines technology, inject it in the individual. Orbital was one of our companies which is doing this for autoimmune diseases, right? But you can do many different things with mRNA. You can do cancer treatment, cancer vaccines now. You can do protein replacement like cystic fibrosis. Arcturus Therapeutics has one where you inhale and cystic fibrosis patients get the correct mRNA. Now, a big part of this space, which you may think that's now well established, that's not true. It's still being developed, right? Is that many of these mRNAs are not optimized for generating the most amount of protein for RNA species that are put in the individual. And so why do you care? Well, you care because if you can get more efficient translation, then I don't have to put so much RNA into you, right? You put a little bit, and that generates the same level of proteins you need. Also, it reduces manufacturing costs. I don't have to make lots of RNA, reduce my cost. Okay, and we know now that the higher protein levels generates higher antibody titer for vaccines and other things, but you can generate basically more beneficial outcome. And making more RNA is not the solution. You get more immunogenicity, you get sort of more side effects, right? But generating more protein per RNA is one. So this is led by Wenhao Jin, a platform leader at Sanford Labs, to try to decipher mRNA translation. Give me an mRNA, can you make more protein from it? Again, quite simple. And the way you measure translation efficiency so far, these are the traditional methods using ribosome profiling. Basically, all your mRNAs in your cells right now you're sitting on have ribosomes sitting on it and translating so you can make protein. So there's a way to measure how much ribosomes, how many there are on every mRNA, and we have, the field has been doing this for many years now, and we have developed some new approaches. So what are we leveraging NAIR for, right? We're here to take first publicly available ribosome or proteomic data sets, ribosome profiling and proteomic data sets, and then we're feeding this into a version of the mRNA large language model I spoke to you about, and then fine-tuning it, and so that we can, given an RNA sequence in some cell type, predict what can we do to make it translate more. A very, very simple problem. So we started initially with a couple of cell lines. So these are cell lines you can grow in the lab readily. You can generate ribosome profiling data, blah, blah, blah. But with the NAIR proposal, we want to expand it. We've actually already expanded this to many, many cell types. This matters, right? Because when you want to make a mRNA for CAR T, you want to go into T cells. You don't want to go in the liver. You don't want to translate in the liver. If you want to do something in the brain, you don't want it to maybe accumulate in the kidney, right? So having a large variety of cell types matters for mRNA medicines and delivery. And so on the y-axis is basically some measurement of performance I wanted to go into. But basically, this pan-tissue data set, leveraging NAIR's resources, and using that to fine-tune the large language model, exhibit amazing performance. They're really quite state-of-the-art. And performance is robust. Like all tissues are working well, right? So it's not like some tissues you just cannot predict. And now we have also put in new experimental paradigms to measure translation at the single cell level. So this is the RiboStem technology from my lab that was recently out in Nature a couple of weeks ago, where we can measure translation for every RNA at single cell level, right, and for millions of cells, right? And now we're leveraging this initially to look at cell lines, bulk cells, but feeding that into the model. And now the model hasn't been trained on this, right? But it turns out it can actually predict translational signals very well compared to RiboStem data sets. It was initially trained in the ribosome profiling data. So basically, just to keep things moving, we are trying to go into a cell type specific way to try to generate mRNA cargo for different cell types. This is still early days, but there's a lot of excitement in the field. There are companies that are being started around in this area. Okay, I'll just end with one, like my second, the last slide, all right, is that I hope you think, right, a different perspective of RNA now. They're never naked. We leverage this understanding of ribosome interactions, RNA-binding protein interactions to build new biology rules as well as therapeutics. And since I'm from San Diego, this is my San Diego beach with my RNAs sitting on the beach, clothed with their swimsuits, I guess. This is also by ChatGPT. And thank my lab, my organizations, and you for your attention. Thank you. And I think I have time for questions, it turns out.
We've got quite a bit of time for questions, actually. So again, if folks have questions, you're welcome to line up at any of the aisle microphones. And if it works better for you for us to bring your microphone, just raise your hand big, and we'll get to those questions. Those are some of the most adorable RNA-binding proteins I've ever seen. And as a former RNA biophysicist, like, I speak with some authority. They're very adorable.
My 5-year-old twins, they love these images. So this is how they think RNA looks like now, right? Yeah.
And thank you for the talk. I just wanted to clarify some of the things you said. It's really not an LLM, right? You're creating a new foundational model using RNA sequence instead of language, natural language.
That's right.
To create a new foundational model and then fine-tune it with specific examples so that it addresses the specific questions you have. So it may be better to call it a new foundational model than LLMs, because people think of LLMs as GPTs and Llama and all these things, right?
We were avoiding the word foundation model for editors of journals. for some other stuff I can tell you about. So, but I agree with you. Yes, I'm nodding my head.
Yeah, okay, great. So how big is your model? Because the kinds of resources you need from there will be based on how many parameters you have. I doubt you have trillions of parameters. Maybe you have billions of parameters.
Yes.
So can you say something about the architecture and how many parameters you're
I think for the first one, we have about 12 to 13 billion parameters. And I think we are leveraging NAIR for part of it. I don't exactly remember. If you ask Karen in the group. And the architecture right now for the large context windows are leveraging the Hyena operator, which has been used for genome sequences.
Okay.
Yeah.
Great, thank you.
Yeah, you're welcome.
You were next, behind Peter.
Hi, my name is Christopher Stewart from Ohio State. And pardon any naivete. My question is about the inference side, right? So you build this model, and I understand how you can understand fundamental biology. When you actually go to apply that, how much does personal variation matter, and how much are we going to have to sample from specific populations in order to get something that's accurate?
That's a great question, right? So as you build this model, we're leveraging, we're basically leveraging a small set of data sets, small number of data sets, sorry, from wild-type individuals, right? So your question is, how would that relate to other, right, patients and so on? So with the ClinVar predictions, it's able to predict the impact of a variation on at least synapse localization, right? But there's so many things that can go wrong when you have a mutation somewhere in an RNA. And I think we and others are looking at how do you find ways to, basically bin that mutation affects this or that on that step. And so right now the field is using wild-type individuals as like a base. This is like, this individual has, as far as you know, nothing wrong with that specific RNA. Learn from that. So then variations may matter. But, I mean, with more resources, we could then fine-tune on patient-specific data sets, right? And I think that's where I feel like life sciences should go, because you get the models to be much more intelligent with better data sets. So that's hopefully coming, just a resource allocation issue.
All right, next two questions over here.
Hi, I'm Mike Viana from AE Studio. Thanks for the great talk. I don't know anything about RNA, but in language models, there's compute optimal scaling laws. So I think it's something like 28 tokens per parameter, as people have discovered. I'm curious if you know, is there something similar for RNA? Is it the same number, if you happen to know, or does it scale differently? And if there isn't a scaling law, how did you choose the number of parameters and versus number of data?
Yeah, that's a good question. I don't know if it's, it should scale this less than proteins, because amino acids, you have more of them, right? And RNA, you have like 4 bases. But we have far more sequences as input. So that's a good question. I would have to get back to you on like the specific details. Yeah.
Okay, thanks.
Thanks, Dr. Yao. My name is Wei Ming from Northwestern University. My question is, have you considered the DA foundation model for your eye domain? This is based on 2 points. One is existing DA foundation model can handle very long contexts, like 1 million base pairs. And the second part is based on the central dogma. DA foundation model should also encode the eye information. So.
Sorry, what's the second question again?
Based on these 2 points, do you consider how well the DA foundation model can perform in your domain? You can only change 1 nucleotide from the output or the input of the DA.
That's right. Yeah, so the DNA, so our RNA model, foundation model, and the DNA foundation models, they all are able to unmask the next nucleotide as you go down, right? And I think our perplexity score for this RNA model is like 2.1, which is I think pretty good compared to other foundation models. The up and downside of the DNA foundation models is it gets very good at predicting things like splicing. Right? Where you remove introns and you can identify splice sites, right? Because in mRNA models, we don't have splice sites. It's like the introns are removed and we're splicing them together. But in the DNA foundation models, so far they've been pretty good at that and transcription control, regulatory elements there, but not so much the mRNA life cycle. And to be fair, I don't really know why. Perhaps they're just not fine-tuning the right data sets. So we are comparing both. We always get that asked from reviewers, but it's something that it's ongoing research. It's a good question. Thank you.
Okay, thank you.
Thank you.
All right, next two questions are back on the other aisle.
Hi, Mark Albers, Mass General Hospital. Thanks for the beautiful talk. I was wondering, some of your motifs, do you think that they're impacting the protein, the ribosome, the RNA binding proteins, and that's what's causing the mislocalization within the neural cells?
Yeah, so we think that the motifs are in a given RNA, and that RNA is therefore you're leveraging that RNA binding protein. So I sort of glossed it, right? But there are specific RBPs that recognize specific sequences, and so we think the RBPs are governing them there. So in our work, I didn't have time to present, right? We have knocked out some of these proteins and seen changes that make sense, that some RNAs are no longer localized. We've also made mutations in some of these sequences on the RNA, and I think that changes localization as well. We've also done things like more biochemistry, like you pull down the RNA binding protein, ask what RNAs are bound at that level of resolution, which we have spent a decade doing. That also has been positive for some RBPs, and we haven't done all of them yet. So I think we're still we're just scratching the surface of this, right? But I think we're getting to the point where we're given a sequence. If you have a variant, we can predict that if it's going to mess up its localization in the synapse. I think that's true. We don't really know always why, but that's pretty accurate now. And I think the fact that from the ClinVar data sets you can predict, a lot of these neurological diseases that are showing up enriched is actually quite exciting. Yeah.
And just to follow, I was curious if you've been able to, in your epilepsy hits, if you've been able to validate that in human brain tissue or not. It could be challenging because those cells, those neurons may die. From the pathologic ones, cells may die with the seizures. But I'm just curious if you
not yet
validate that.
Not yet. Good question.
Thank you.
Good question.
I see you made use of INR operator, which would allow you to undo really long RNA sequence. So my question is, for the model, what is the maximum base pair sequence that the model can undo? Yeah. And also at inference stage, for an 80 GB A100, what is the maximum base pair that can fit in? And also, secondly, on the ClinVar data set, do you also try to see how these mutation can affect the pathogenicity?
Right, great questions. The input context window is about 8 KB, 7 to 8 KB. And so that is the length of the majority of mRNAs, not pre-mRNAs. And for the inference, my recollection is that we take the same input size. But when we do occlusion mapping, we're taking, the whole thing and then basically making mutations. So in silico, so we make the mutations that overlap with the ClinVar data sets, and where that predicts that it doesn't localize or not, then we call that a interesting hit that then we overlap with what the ClinVar phenotypes are. But we haven't, so hopefully that answers that part of the question.
Pathogenicity, if it's benign or not.
So we don't fully know, right? So we are saying, okay, of all these neurological diseases, or at least the ones that came up, we think the variants are affecting where the RNAs are in synapses, but we don't know if that's true. Basically, we're giving a whole set of new hypotheses to test, right, for these different diseases that, hey, that mRNA with that variant, it's what's causing epileptic seizures, but we don't know that, right? And so I think we're starting to do, so part of my lab does a lot of iPS stem cell models for neurological diseases, and we're starting to make models of neuronal excitability and cell tissue culture, make the mutation, hopefully we can see the phenotype. But that's probably another year or so. But it's kind of exactly where we're going to. Yeah.
Okay, thank you.
Thanks for asking the question. Maybe wasn't clear in my talk.
And I think we've got time for at least the next 2 questions here, maybe the next 3.
Okay, I'll just ask my first question then. John May, USC, thank you for the nice talk. So when you put up the architecture slide in your first part, you said it was just a feed-forward model?
Yeah.
So there's no attention? You're not using transformer architecture?
I don't think so. Yeah.
I'm wondering about that decision. I would think that the attention would be helpful.
Yes,
also, I guess I'm wondering how, thus you made a 12 billion parameter, just a single feed forward model that just took in an 8K window to predict one next chemical?
Let me check with you on whether we had included transformers in there. It's been a while since I've looked at the initial architecture.
Is there a publication?
It should be on Bar Archive in a couple of weeks. But yeah, I mean, this was started like maybe 5+ years ago. I don't quite remember.
Cool, thanks.
Thanks.
Thank you for the very entertaining and engaging talk. I mean, we're all lining up to ask questions, so it sparked interest, right? I have a question about the AUC curves that you showed in the first part.
Right.
If I saw it correctly, on the left was for animals and on the right for humans.
Oh yeah, mouse on the left. I mean, on your left.
Mice and humans, right? And
yeah.
I think I noticed that the AUC for humans was a bit lower than for mice.
Yes.
Do you know why that is and what we would need to close that gap?
So the mouse data sets were, again, if I recall right, I think 3 different tissues in the brain of mice isolated for synaptic RNAs. The foundation model was primarily trained on that data set. There was no training on the human data set. So it was just basically run on the human sample, like the isolated RNAs, there were synapse or not, and the AUC was generated on that prediction. So there might be differences in maybe conservation between species. There might be because the human neuronal data set was generated from cell culture models, not like a human brain sample. So maybe there's this loss of translation in a plastic dish, right? But the fact that we still get a pretty good. It's trained only on mouse tissue. It's pretty good. I mean, we were actually quite surprised.
So is training on data from human brains something that would be possible in the future?
We need to figure out, but I don't think we've got good synaptic RNAs from brain samples from humans yet. I mean, we should go take a look and see. But I think there's a high level of conservation, at least because the foundation model trained on the mouse brain seemed to predict well on the human iPS-derived neurons, right? So I think we're going with that for now. But of course, I mean, there's probably many more ways to enrich the data sets. Yeah.
It's impressive. Thank you.
Yeah, I have a very brief question. I appreciated your presentation a lot as well. In my very limited knowledge of drug discovery, I know that one of the issues in timeliness that is faced is ruling out non-viable candidates. Does your model assist in that as far as that goes?
Yeah. So I think it's quite well understood now that one of the biggest bottlenecks to getting new drugs is not the chemistry or not so much manufacturing trauma. It's like getting completely new targets, right? And the problem is completely new, like targets are not proprietary, right? So all pharma companies are hitting the same targets with different strategies, but getting a new target then validated, the problem is that takes takes more time, right? But I think that's a big bottleneck in the industry where like everyone's hitting the same, like there are hundreds of companies hitting the exact same thing, sometimes with the exact same strategy, right? So new targets are missing. So I think our work here will tell us that a specific RNA target might be a new target for epileptic seizures, maybe a new target for neurodegeneration, right? So it's all, at this point, a lot of these are now becomes hypothesis generating work, right? So that's, there's not a small molecule drug yet, but.
Thank you.
Yeah. Thank you.
So I've got one last question, but I think it's a really quick one. The acting NSF director mentioned that it might be very awesome to have something like an AlphaFold across scientific domains. Is there the potential for a big AlphaFold discovery for the RNA world, or is the nature of RNA mean that it would really take multiple of those?
Yeah.
What do you think?
I think it's a good question, right? So remember, AlphaFold is based off of protein structures for protein, right? Collected 30, 40, like many years, many labs, right? If you go and ask, is there an equivalent data set for RNA structure? There isn't, right? And if you ask, okay, even if there are some RNA structures that are available, do they actually represent what's in the cell? The answer is no, because they're all naked as a structure. In the cell, they are probably complex with different proteins, right? So the question is, are there protein RNA complexes structures, right? Not really. So if you have, not enough, right? I think Riju Das at Stanford and others are now trying to collect this kind of data set. But I think it needs the whole community to get into there. Then you can get an AlphaFold for RNA. I think that works, right? But I think right now, in my opinion, right, the field is not lacking computational expertise. It's lacking good data sets for the right biology.
Thank you.
Okay.
And let's thank Jean again.